Assalamualaikum and good morning everyone. My name is Noishika and for our first practical lab, we are studying the effect on environmental factors on the role of microorganisms. The main goal of this lab is to see how different physical and chemical conditions act as limiters for bacterial life. We are focusing on four major factors, which is temperature, osmotic pressure, pH level, and oxygen availability. As we know, every microbe has a comfort zone where it grows best. By testing four different species, Pseudomonas aeruginosa, Bacillus subtilis, Staphylococcus aureus, and E. coli, we can observe which bacteria are more resistant and how changing these factors can either encourage their growth or stop it internally. Now, let's look at our material and method.
In this experiment, a pure culture of microorganisms was used and inoculated into Diabeticus soy broth, also called as TSP, to study the effect of environmental factors on microbial growth. For temperature analysis, 1.1 ml of the culture was added into 4 tubes, and it was incubated at 25, 30, and 50 degrees Celsius for 44 hours without shaking. For osmotic pressure, the culture was inoculated into TSP containing different concentrations of salt and sugar, and also incubated at 37 degrees Celsius. To evaluate the effect of pH, the culture was inoculated into TSP, adjusted to pH 3, 5, 7, and 10, and also incubated at 37 degrees Celsius. For oxygen recruitment, one tube was overlaid with paraffin oil to create anaerobic conditions, while the other tube was exposed to oxygen to allow aerobic growth.
Now I will talk on discussion part of our lab report. Based on the results obtained, we have successfully identified on how the temperature, pH, salt, sugar, and oxygen factor affects the growth of E. coli. Then through this discussion, we have also able to compare the effect of temperature, pH, salt, sugar, and oxygen on E. coli and other microorganisms such as S. aureus. Based on the results, at 5 degrees Celsius, the E. coli did not show growth due to low metabolism. And based on Science Direct, the other microorganism did also not show growth at 5 degrees Celsius, and the reason was the same, where the metabolism was too slow. 25 degrees Celsius, the growth of E. coli and S. aureus is minimal, and therefore the turbidity level is moderate for both. 37 degrees Celsius is the most optimum temperature to support the growth of E. coli and S. aureus at a maximum rate. Temperature of 55 degrees Celsius causes the E. coli to die because of protein denaturation, and S. aureus can resist the temperature and can show little growth due to thick cell wall resisting the heat. Temperature of 55 degrees Celsius causes the E. coli to die because of protein denaturation, and S. aureus can resist the temperature and can show little growth due to thick cell wall resisting the heat. Next is pH factor. The pH value of 3 is acidic, causing the E. coli and S. aureus to not survive as the enzymes and protein will denature. Therefore, turbidity did not occur. At a pH of 5 that is slightly acidic, the both E. coli and S. aureus showed little growth due to adaptive mechanism. At a pH of 7, both E. coli and S. aureus shows good turbidity and growth because the pH is optimum, supporting the enzymatic activity and metabolism. At a pH of 10, both S. aureus and E. coli did not show growth and did not show turbidity because the pH was extreme, destroying the protein. The next factor is salt concentration. At 0%, the E. coli and S. aureus has grown well because there was no salt and dehydration did not occur. Then at a salt concentration of 1%, the growth of E. coli and S. aureus is still good and showed good turbidity because the salt was just very little and did not cause dehydration. Then at a salt concentration of 5%, the E. coli did not show growth due to dehydration. However, S. aureus has showed little growth because it is cellular tolerant and has adaptive mechanism. At a salt concentration of 10%, both S. aureus and E. coli did not show growth because the dehydration was extreme. Next factor is sugar concentration, which gives the same effect as salt concentration. At 0%, both E. coli and S. aureus will grow and shows good turbidity. 5%, the E. coli don't grow due to dehydration and S. aureus grow due to adaptive mechanism. At 10%, the both E. coli and S. aureus does not grow due to extreme dehydration. Next factor is oxygen. The E. coli in broth with paraffin oil showed little growth because E. coli is facultative and it grows below the oil layer. And without paraffin oil, the growth of E. coli is good and turbidity is also good. For S. aureus, the growth is same as E. coli, where with paraffin oil, the growth is below the oil layer, and without the paraffin oil, the growth is good.
Hi, so I will continue for result part. For temperature, tubes different in color intensity across temperature range. This is because enzymes and microbes have an optimal temperature where activity is highest, which is strongest color. If color is too low, mean reaction slow down. If the color too high, protein denature, microbes die, leading to weak or no color change. The pattern likely shows peak activity at moderate temperature. So next, oxygen. Tubes show differences between oxygen-rich and oxygen-limited condition. This is because aerobic organisms thrive with oxygen, producing strong color change. Anaerobic organisms or facultative anaerobe may still grow with the oxygen, but with different metabolic pathway, often weaker or different color. The contrast shows how oxygen availability shift metabolic activity. For sugar, tubes under sugar condition show distinct color change with darker tone in some. This is because sugar serves as an substrate, which is food source for microbes or enzyme. Higher sugar concentrations usually enhance activity, producing more metabolic byproduct like acid or pigment, hence stronger color. Extremely high sugar can also cause osmotic stress, slowing growth, so they are balanced. For salt, test tube show varying intensities of color depending on salt concentration. Some appear lighter, other darker. So, this is because high salt concentration often inhibit microbial or enzymatic activity due to osmotic stress, cell lost water. Lower salt level allow more growth or reaction, leading to stronger color change. The gradient suggests that salt acts as a limiting factor. Too much suppresses activity. Moderate levels allow optimal reaction. Okay, for the last one, which is pH. Tubes vary from light to dark depending on acidity or alkalinity. This is because microbial growth and enzyme activity are highly pH dependent. Strong color change at neutral or slightly acidic pH suggests optimal condition. Extreme acidic or alkaline conditions reduce activity, leading to pale or no color. The color differences may also directly reflect pH indicator reacting to metabolic product. For conclusion, this study demonstrates that E. coli growth is highly sensitive to environmental conditions, with optimal activity occurring under moderate salt and sugar concentration, neutral pH, and mesophilic temperature around 37 degrees Celsius. Oxygen availability further influence metabolic pathway, with aerobic respiration producing stronger growth compared to anaerobic fermentation. The observed color change in the test tube serve as a reliable indicator of metabolic activity, highlighting how stress factors such as osmotic imbalance, extreme pH, or unsuitable temperature can inhibit growth. Overall, the finding emphasizes that the importance of balanced environmental conditions for sustaining E. coli activity and illustrate its versatility as an facultative and aerob capable of adapting to diverse habitats. So as you can see, this is the differences that we use for this lab report. That's all from us. Thank you.